I'm Tae Kim from Process Development Downstream Process team here at Samsung Biologics. In the fast evolving world of biologics manufacturing, the stakes are higher than ever. Therapies are becoming more expensive, regulatory expectations are rising, and patients are waiting for effective medicines with speed and precision. Today, we will dive into a powerful yet often underestimated tool that is transforming how manufacturers achieve these goals. Small-scale downstream process studies. These studies are critical enabler of tech transfer, process validation, and regulatory filing. And in this session, I'll guide you through how this could be achieved in the small-scale studies. The development and manufacturing of biologics is a complex and lengthy process. As you can see, it starts with basic research, progresses through drug discovery, preclinical studies, clinical studies, FDA review, and finally, post-approval research and monitoring, or phase 4. Each phase presents its own set of challenges, and the complexity increases as we move toward commercialization. So what drives complexity? Molecular complexity or diversity expansion beyond monoclonal into bispecific, fusion proteins, enzymes, mRNA, peptides, and more. Evolving regulatory expectations, agents are requiring deeper process understanding and robust data-driven control strategies. Operation efficiency pressures, faster timelines, and cost constraints are demanding smarter process designs. As the industry challenges evolve, manufacturers need to integrate rigorous control strategies and enhanced process understanding, which are essential to ensure the reliability of biomanufacturing. So, what exactly are small-scale downstream studies, and how can they help? Small-scale studies are lab-scale models that simulate full-scale manufacturing conditions. These studies provide predictive insights into process performance and risks, allowing for faster decision-making while conserving valuable time, cost, and resources. Roles of small-scale downstream studies in scaling up include, but are not limited to, verification runs, filter sizing, and investigational studies. And in scaling down, process characterization, resin membrane lifetime, virus clearance, and so on. Small-scale downstream studies are no longer confined to early development. Today, they're essential across the entire product lifecycle, supporting tech transfer, process validation, and regulatory filing. They serve as a predictive tools, decision-making accelerators, and safeguards against risk before they impact large-scale production. The studies thus offer manufacturers a flexible and cost-effective way to interrogate process behavior, ability to identify risk before full-scale production, and reliable data that strengthens regulatory confidence. During the tech transfer, they confirm reproducibility and de-risk scale-up. In process validation, they generate the scientific rationale for control strategies. For regulatory filings, they provided targeted data to address agents' queries. Through all these applications, they bridge scientific understanding with regulatory and operational excellence. Tech transfer is the process of transmitting product and process knowledge between development and manufacturing sites. There are several key success factors that contribute to optimized tech transfer. At Samsung Biologics, we offer 3 key traits to facilitate tech transfer, which are proven expertise to ensure on-time tech transfer, maximized efficiency in executing phase-appropriate tech transfers, and additional support to guide throughout its entire lifecycle, including process characterization and process validations. Small-scale verification runs are performed prior to manufacturing at scale, starting from USP or upstream process cell culture and downstream process DSP purification to the analytical test method transfer, and though not part of tech transfer, but in a later stage, ending with process characterization and process validation. Small-scale studies support the process and ensure preliminary gap assessment that identifies potential discrepancies between the sending and receiving units. As discussed briefly in the previous slide, successful tech transfer relies on several factors. Real-time communication by building a strong collaborative environment, seamless knowledge flow between sending and receiving units. Together, these ensure that no surprise occur at any scale. Ensuring process reproducibility and product quality by deep understanding of process, knowing the product and critical process parameters. Facility fit and risk management, aligning the process with the receiving site equipment and capabilities, and anticipating potential process issues or deviation and their corresponding mitigation plan. And finally, data-driven insight, leveraging small-scale studies and analytical tools to validate assumptions, de-risk scale-up, and support evidence-based decision-making. These four core principles are interconnected in a circle for successful tech transfer. So verification runs are critical checkpoint in tech transfer. They're small-scale execution of transferred process designed to confirm that the process performs as expected at the receiving facility. Think of them as the bridge that ensures smooth tech transition from development to commercial manufacturing. The primary purpose of verification runs is threefold: confirm reproducibility of process performance and product quality across sites, evaluate gaps between sending and receiving units, de-risk scale up before moving to scale. These allow us to validate assumption with data long before we commit to manufacturing by comparative analysis against historical standing unit data to ensure alignment in process performance and product quality. Results across different modalities, including monoclonal, fusion proteins, and enzymes consistently aligned with the historical performance data provided by the clients. These demonstrate that the verification runs are not limited to one modality, they are broadly applicable across biologics. In short, verification runs act as an early safeguard, making tech transfer more predictable and reliable. Filter sizing tests are also an essential part of downstream tech transfer. They allow us to design scalable, efficient, and cost-effective filtration strategies while ensuring consistent product quality. Main objective of filter sizing tests are to select suitable filter types and pore sizes, define optimal operating conditions, prevent oversizing, anticipate fouling risk before appearing at manufacturing site. The obtained data from the small scale is typically linearly scalable to manufacturing. Such studies are applicable to any filter that is used during the process, including dead-end or normal flow filtration used for depth and pool filtration, and tangential flow filtration, TFF, used for ultrafiltration and diafiltration. Results from each of the modes, from monoclonal, bispecific, fusion, and enzymes in two distinct filtration process stages were normalized against a client-provided filter capacity by 100% and showed comparable results in our site. When process issues or deviations occur in the manufacturing, we need a way to respond quickly without disrupting operations. Investigational studies provide that capacity. They replicate manufacturing scale conditions at lab scale, allowing us to troubleshoot efficiently and accurately. Given challenges, but not limited to on the left, such as low product yield, unexpected filter fouling, inconsistent impurity clearance, the investigational studies follow a structured approach by simulating manufacturing scale issue at lab scale, conducting targeted experiments using a representative model, applying advanced analytics to uncover root causes, and if required, statistical tools such as design of experiments to systematically test input-output interactions. These combinations ensure robust data-driven troubleshooting that eventually helps us to set up corrective and preventive action, CAPA, thereby enhancing long-term process reliability. Process validation is a cornerstone of biologics manufacturing. It aims to ensure that every batch produced meets quality standards consistently. In order to achieve this goal, validation requires deep process understanding, robust and rigorous control strategies based on scientific rationale, and thorough documentation. While full-scale validation is the goal, small-scale downstream studies generate scientific rationale needed for developing control strategies, identifying and mitigating risks, simulating at-scale operations under controlled laboratory conditions. This approach conserves both time and resources while ensuring processes are well understood before commercial execution. The offered small-scale studies as SBL are process characterization, resin and membrane lifetime study, buffer and intermediate hold study, reprocessing study, viral clearance. We are always also open for any other regulatory that support experimentation. Process characterization study is a backbone of validation as it identifies how process parameters affect product quality. Beginning with risk assessment, such as failure mode and effective analysis to identify key parameters, input-output relationships are studied by statistical design, quality by design or QBD, to establish proven acceptable ranges, PAR, for critical parameters. To put it simply, process characterization is a systematic approach to understanding and controlling process variability through statistical design for PAR establishment. We can define the design space, which is the range of operating parameters that ensure consistent product quality. The data from small scale helps to identify what parameters have a significant impact on key performance indicator or KPI or critical quality attributes CQA, leading to manufacturing control strategies based on enhanced process understanding. In the figure on the right, the boundary of the colored areas represent the lower or upper boundary of quality attributes in terms of process parameter X1 in x-axis, process parameter 2, X2 in y-axis. The uncolored area, or the white area, in this case represents consistent quality, and the dotted red line means proven acceptable ranges for X1 and X2, each represented by its lower and upper limit. Resin membranes are critical for purification as well. Their lifetimes must be validated to ensure consistent quality and efficient across the product lifecycle. Small-scale resin and membrane lifecycle studies are then utilized for determining the maximum number of cycles and cleaning adequacy and help to evaluate performance over repeated cycles and use that to extrapolate to manufacturing scale. The trends in performance and product quality can be tracked during lifetime studies to determine their usable or viable lifetime and assess the adequacy of cleaning based on the product carryover. Maximum number of cycles that are allowable for the resins and membranes can be determined based on these results and can be linearly extrapolated to manufacturing to serve as a critical reference for process control, product purity, yield, and impurity clearance. As a case study in the figure, 3 intersections are reached for impurity 1, impurity 2, and key performance indicator KPI 1. Among the 3 interactions, the earliest interaction, which is the upper limit of impurity 2, is determined to be the maximum allowable cycle for either resin or membrane. The purpose of hold time studies is to also examine the stability of buffers and intermediate process materials over defined storage durations. These studies simulate worst-case manufacturing scenarios where samples at predefined time points are monitored to determine the time point at which the material is no longer meets acceptance criteria. From this, the maximum allowable hold time can be established. These results will help optimize production scheduling, support regulatory filings, and provide confidence in the process consistency. Similar to the previous case study, purities and impurities of the whole materials are assessed at the predefined time period, and the earliest intersection that meets the upper or lower limit is then determined to be the maximum allowable hold time. A viral clearance study, or VCS, is a regulatory requirement that ensures manufacturing processes effectively remove or inactivate adventitious viruses. Common viral clearance methods include solvent detergent treatment, low pH viral inactivation, chromatography, and virus filtration based on size exclusion mechanisms. The small-scale spiking studies use model viruses to demonstrate clearance across unit operations and is typically conducted in 2 phases: an early stage VCS following IND submission, and a late stage VCS prior to BLA submission. The figure represents a case study for BLA filing where removal of 4 viruses is investigated, along with the downstream process from capture, polishing, low pH virus inactivation, and filtration. Cumulative log reduction values are summarized. As one could see, for low pH VI, which specifically targets enveloped viruses, it had no impact on the non-enveloped viruses. With the downstream processing at the site, acceptable log reductions values could be also achieved. Finally, all of these small-scale studies support our regulatory filings. By expanding manufacturing information with small-scale study data, we can strengthen our BLA submissions and facilitate the approval of new biologics. It allows us to address product or process-specific questions and leverage data from tech transfer and validation. Key elements in a BLA submission include quality control measures, non-clinical data, clinical data, and manufacturing information. Examples include maximum intermediate hold time verification and excipient stability optimization. In conclusion, small-scale downstream studies play a pivotal role in advanced biologics innovation through deep data-driven understanding of manufacturing processes. From early development through commercialization, these studies provide a flexible, efficient way to assess risk, confirm process robustness, and address regulatory expectations. By supporting critical activities such as tech transfer, process validation, filing preparation, they ensure that processes are well characterized and compliant while minimizing cost and complexity. As biologics manufacturing continues to evolve, the strategic application of small-scale studies will remain essential for fostering innovation, ensuring quality, and maintaining flexibility in a dynamic regulatory landscape. And with that, thank you for your time for this webinar. We hope to see this session has provided valuable insights into our PDDSP capabilities. Thank you.
How do verification runs at small scale ensure reproducibility across different facilities, especially when there are equipment or material differences between the sending and receiving sites?
Okay. So verification runs serve as a bridge between development and commercial manufacturing. They begin with the gap assessment, identifying potential discrepancies in equipment design, materials, or operating conditions. Then small-scale experiments replicate the transfer process at the receiving facility. Maybe we are looking into direct comparison against the sending unit's historical data. Even if the differences exist, the verification run provides a data-driven confirmation that the process will be generated at the expected product quality or flags critical gaps that must be resolved before full-scale production.
All right, thanks. Another question for you. When deviations occur during commercial production, how quickly can small-scale investigational studies typically provide actionable insights? And what tools are most effective in root cause analysis?
The speed actually really depends on the complexity of the issue, but the advantage of this investigational study is that they can replicate full-scale problems at lab scale, which allows for rapid iteration. By combining analytics, spectroscopy, and maybe statistical tools such as DOE, teams can systematically test variables and identify root causes in a fraction of time it would take at scale. These approaches can minimize production downtime and support corrective and preventive actions. Kappa?
Great, thanks. One final question for you. Regulators often emphasize process understanding and risk-based approaches. How do small-scale studies like process characterization or viral clearance strengthen regulatory submissions and address agency queries during BLA preparation?
So the small-scale studies, they can provide the scientific rationale regulators are looking for. For example, process characterization study defines design space and proven acceptable ranges for critical parameters and supported by DOE and risk assessment. Similarly, VCS, viral clearance studies, use spiking experiments to demonstrate robust log reduction values across unit operations. These data-driven outputs give regulators confidence that the process is well understood, consistent, and reliable. And they also provide flexibility to answer specific regulatory queries quickly, since small-scale model can generate supplemental data without disrupting manufacturing.